where we left off, the liver lobule. Think of the liver lobule as the structural functional unit of the liver. It's a histological structure which basically gives you these hexagonal lobules of hepatocytes. Liver lobule. You got a hexagon with a little shower drain in the middle there. And the idea is these lobules are packed out with hepatocytes where the blood that it's filtering through so that hepatocytes can do the job of the liver flow towards the sour drain. What's the central vein? And so let's remember that the blood has a dual blood supply. The liver has a dual blood supply. That is, it's got proper hepatic artery, and portal vein. Go back and look at those notes that we took for that. It's the proper hepatic artery that's got the oxygen-rich blood. The portal vein is about, it's oxygen-poor blood, but it's nutrient-laden. And actually, the liver receives about 75-25%. 75% portal blood, about 25% is the oxygen-rich blood from proper hepatic artery. And that's the blood that's flowing towards the shower drain there. It has a singular venous drainage system. Singular venous drainage. And so on this slide, the liver lobule, it's the central vein. All those central veins of each lobule, they all drain to hepatic veins. And it's the hepatic veins that drain directly into the inferior vena cava, IVC. Okay, so two in, one out basically. At each corner, what you're going to have is, as the blood flows towards the shower drain, the hepatocytes have a chance to process all the food you ate and to extract all the oxygen and nutrients. But also, bile produced by the liver flows in the opposite direction towards the corners. And so really the histology, what you should be able to identify is the central vein in the middle, and then at each corner of the hexagon, what's called a portal triad. Not to be confused with porta hepatis, which I taught you last time on the top. The portal triad is this associated with the liver lobule. At each corner, there's three structures you should be able to identify. One is a branch of the hepatic artery. Also, I'll put a little circle representing a branch of hepatic artery at each corner there of the hexagon. There's also a branch of the portal vein at each corner. So I'll use blue because it's oxygen poor blood, but nutrient laden because it came from the intestines. The third structure is the bile canaliculi, a branch of the bile duct basically. So those three things are the contents of what's called a portal triad. Once again, portal triad has these three things: branches of ... portal vein, hepatic artery, and and a branch of bile duct. Okay, so know what a portal triad is. It's those three things. And the blood vessels that flow towards the central vein are called sinusoids.
So it's not proper hepatic arteries, right?
Oh, it's either, and I use both interchangeably. And I think in most keys, older keys, they leave off the proper.
Because
So
I thought you said there is different, right?
I said there's proper hepatic artery, and let me just write it since it's causing confusion. I'll write it every single time, I won't be lazy. Because sometimes students call it hepatic artery, and I give credit for that too. You don't have to put proper. Only in your atlas, I see they put proper hepatic artery. More often it's called just hepatic artery. But I said don't confuse it with portal vein. Okay, I mean, I guess that's the confusion. Any other questions on what proper hepatic artery is? So you can put it or not, it's all good. I guess the main confusion is students call portal vein hepatic vein. Or you don't even know what's what. You just put something because you don't know the material. That's the most common confusion. Hepatic vein versus portal vein. Anyways, I'm going to draw blood vessels going right to the central vein. So this one in the middle there, that's central vein. And then all these are all these things draining. Each of these is a sinusoid. And the sinusoid is a mix of both the oxygen-rich blood flowing towards the shower drain and the portal blood flowing towards the central vein, what I keep calling the shower drain. All right, so sinusoidal blood is a mixture of blood. Of those two things. Okay, so associate this note with this. So now you know what's in the mix. It's 75-25, portal blood, oxygen-rich blood. So that having been said, you got hepatocytes all throughout this structure. There's a picture there. One thing about the design of the sinusoids flowing past the hepatocytes is the arrangement of the hepatocytes is basically in rows of two. So check this out. I kind of blew it up here. Here's a row of two cells here. These cuboidal cells, those are hepatocytes. And as blood flows by, it extracts the oxygen and the nutrients you ate, and they also are stimulated to produce bile. So I'm going to draw the hepatocyte like this. I'll put a little groove there. That's an hepatocyte. I'll draw a few of them in a row here. A little hepatocyte. That's one row. Let me draw another row of four. Two rows of four. That's kind of the arrangement of the hepatocytes. You have this membrane That's, well, like I say on this slide, it's the basolateral membrane. It faces the sinusoidal blood. Basolateral side. The other side, that side, apical. Well, anyways, the side that faces the sinusoidal blood. Maybe blood's coming in. Maybe going this way. So as blood flows by, it's the hepatocytes that have a chance to extract oxygen and nutrients. They even named this gap between the cells and the blood. So this gap is named the space of Dissay. And so you have a sinusoid at the top too. I mean, I won't draw it. You can see on the picture. There's one on the top and the bottom. So by having this arrangement, 2 rows of cells, every cell is exposed to the sinusoidal blood, okay, as it flows towards the central vein. Now, on the in terms of secretion, on the other side, the apical side, you're secreting bile into that little gap. This is called the bile canaliculi. This little space right there. It's a gap between hepatocytes. I'll just describe it as a gap between cells. A gap, not a duct, between cells. Because if you look at the picture, they draw the bile canaliculi as its own separate duct. They use the color green. But my understanding is that that's not the case. It's just a gap between cells. It's not a duct. But eventually it'll drain into a duct. That's certainly a duct at the corner there, a branch of the bile duct. At what point it does it, I'm not sure, but at the cell level, the bile canaliculi is just a gap between cells. Bile itself is composed of bile salts. And you don't have to know them, but here's what you do need to know about these bile salts. Cholic acid, deoxycholic acid is that, well, they have 2 sides. The 2 sides, one is polar, one is non-polar. So one is hydrophilic, and the other side is hydrophobic, and they can interact with the fats to emulsify the fats you're trying to digest. So that's really the main action of bile I want you to know, fat emulsification. So let me erase this here. So bile salt, with these salts there. So in this artist's rendition of bile salt, the idea is one side, it kind of faces the fat. Because it's hydrophobic, and the bile salt side faces the watery lumen of the digestive tract. Faces water. And by doing that, you accomplish, I'll say bile salts accomplish fat emulsification. Fat emulsification aids in the digestion process. It's not fat digestion, but it helps it, because what you're doing is you're exposing more triglycerides to the lipase activity. expose more TGs, that's triglycerides, to lipase activity. And by doing that, well, you're breaking big fat globules down into smaller fat droplets, and that makes them easier to absorb by the lacteal. That's what's shown in this picture here. So this picture, bile salts break down a large fat globule into a bunch of smaller fat droplets, I guess. Making them smaller, you're, again, number one, you expose more triglycerides to lipase. And making them smaller allows lacteals to absorb fat into circulation. The lacteals at the bottom of the slide, and that's kind of what you see, the fat absorption there into the villi of the small intestine. Here's true fat digestion. Here's lipase cleaving off the free fatty acids. So that's what the lipase activity does. It's the fatty acid. They're hydrocarbon chains. They can enter the cell and that's what you burn for energy. So lipase activity, I'll put it under this. You're increasing free fatty acids. They're just circulating more. All right, so let's talk about the small intestine. This happens in the small intestine. So the anatomy of the small intestine, there's not much to look at. It's just the tube that's 21 feet long. It's about an inch in diameter. And I already talked about the duodenum. That's the first 10 inches. This picture shows you jejunum ileum. We teach them as 3 different things, but there's just one continuous tube. It's about 21 feet long, about an inch in diameter. It starts here at the duodenum, then it goes jejunum, then ileum. And you are expected to know the difference. We usually tag it kind of at the juncture, so the end of one, the beginning of the other. So if I tag it way down here, it's the end of the ileum, the beginning of the ascending colon. Okay. It's greatly coiled. That's what it looks like, greatly coiled inside the body cavity. So here's a side-by-side so you can kind of see where they cut it away from its mesentery. Remember I gave the analogy of the small intestines being a python in a cloth bag, and if you gather up the cloth bag, that's like the root of the mesentery that holds it to the back body wall, and they cut it right there and right there. So I know that's the beginning of jejunum, and that's the end of the ileum. So for small intestine, the main function is absorption. That's the main job. But it'll finish off digestion should there be anything left to do, because there's brush border enzymes. So the anatomy is recognize duodenum, jejunum, ileum, just to write them out, just those three. All total 21 feet, that's length. About 1 inch diameter. There's small intestine, there's large intestine. The small intestine is longer, but the width is smaller. So that's why it's the small intestine. The main job is absorption, and it's modified well for absorption. What I want you to know are modifications to increase surface area. Because if you have more surface area, well, you can accomplish absorption better. More area to absorb food. And, well, I'll just kind of fly through this. It's plica, it's villi, and it's microvilli. Okay. Now the plica, you can see with the naked eye. When you start to get the villi, microvilli, then you start to need a microscope. All right. So I think the picture will describe it better than my slide here. The picture shows the first couple of levels of surface area modifications, the plica, which are called circular folds on this slide, and the villi, these little finger-like projections that are much smaller. You can only really see those better with a microscope. And so, if you like look at a close-up of here, I'm going to draw this red line here. You can see how folded it is. So let's remember our layers of the GI tract wall: mucosa, submucosa. The other layers aren't shown here. So I'll draw this last one here, this big old plica. Muscularis mucosa. Now, if you put kind of the epithelium over it, it's all like that wavy villi stuff. So deep to the muscularis mucosa, you have the loose connective tissue of the submucosa. So let me put that. So this is loose, CT of submucosa. Now notice that it goes all up into here, into this plica fold. And well, what we see is that this whole big fold sticking up is plica. The full name is plica circularis. Sometimes they're just called circular folds. I'm going to call them plica because I like that better. And what you see is that plica is a folding of the submucosa, right? Because it goes all up in it. Let's see here. Folding up submucosa. Now, just deep to the villi, these little things I'm drawing here, those are villi. That's the epithelium, the black line. But the connective tissue just under is the lamina propria, the LP. So I'll just color part of this green. I don't want to color the whole thing, but I think you get the idea of what this is. So that's, the green is connective tissue. It's lamina propria. So So the villi are a folding of the mucosa. You know, if you could look at an few individual villi, you would see that they're lined with a columnar epithelium. And if you could just look at one or two epithelial cells, the microvilli brush border is the final smallest level of folding to increase absorptive area. There's a cell. Microvilli. Sometimes it's called quote unquote brush border because that's how it appears under histology slides, and they're studded with the brush border enzymes that I mentioned earlier that finish off the digestion. I'll just draw a few little red dots to symbolize the brush border enzymes. Well, anyways, the microvilli are a folding of the cell membrane, okay? And that's the last kind of modification to increase the surface area. So I'll squeeze this in on the side. I'm writing folding of the plasma membrane. Put PM, plasma membrane. So you fold at the mucosa, submucosa, and plasma membrane levels. There's a picture of the small intestine model we looked at before, so you can kind of study those to practice the structures. Now the colon, once we get to the end of the small intestine, duodenum, jejunum, ileum, if we look at the colon isolated, that's a good picture to study. It has all the components of the large intestine. Large intestine. Colon. You know, by the time food reaches here, there's not much left to absorb. Maybe just water, and then you compact the food stuff into fecal matter. Compacts food stuff. Feces. Well, the structures include, I'll start at the beginning. For food stuff to enter from ileum to the first part of the large intestine, it's got to go through those little lips there. I call that the ileocecal valve. They call it like ileocecal labrum superior inferior lips, same thing. It's like a valve. The lips are the valve. So the structures include ileocecal valve. That's how the food stuff gets into the first part of the large intestine, which is the cecum. It's that little pouch on the bottom there. Cecum. And hanging off that little pouch is an appendage called the vermiform appendix. I misspelled it here. That says veriform. This is the correct spelling here. Vermiform, which means worm shape. Vermiform appendix. This is the same appendix I always hear has to be removed in emergency surgery. If it becomes clogged with fecal matter, it can block off the blood flow. That will cause pain, and well, if it kind of like bursts, that'll cause a lot of problems. It is an emergency surgery. You want to remove it before it perforates, basically. Okay, well, from the vermiform appendix, hanging off there, the cecum, what happens is the longitudinal layer of the muscularis externa, it condenses into 3 little bands that run up longitudinally. Now you can't see the formation of it because they cut away so you can see inside there. So from the cecum, the longitudinal layer form these ribbons called the tenia coli, right there. They're longitudinal bands of smooth muscle. Tenia coli, there's 3 of them, and you can see maybe one here. 3 ribbon-like structures. So again, the longitudinal layer of, I'll just put ME for muscularis externa. It's reduced to these 3 band-like structures. Reduced to these band-like structures. So when they can contract longitudinally, it's kind of like a drawstring, you know, bunch it up, and it creates that pocketed appearance of the large intestine, which is another visual difference between large and small. The large intestine, those pockets are called haustra. Haustra. Pocketed appearance of large intestine. So I'll talk about the large intestine in terms of how you pass through it. First, you go up through the ascending colon. It's the first 12 to 20 centimeters. It's about a foot long, and well, basically it's retroperitoneal. Now, so the intestines were intra, so this ascending colon is mostly retro. So ascending colon. I'll just put retro for retroperitoneal. Things alternate intra-retro. I mean, they have to. You're either intra or retro. You can't be both. You're one or the other. So it kind of alternates. The stomach is intra. The duodenum is retro. Jejunum ileum, those are intra. I'm going to go back retro. Now this ascending colon, it stops when it gets to the top, which is, we can't see it. It'll stop when it gets to the bottom of the liver. Right there. The liver, it takes a turn. It's called the right colic flexure because that's where the liver is, so you can also call it the hepatic flexure. I'll just list it as a term. Right colic flexure. You can call it hepatic flexure if you want. Just remember, liver on the right, spleen on the left. When you make that turn, you go into the transverse colon, which has that looped down experience. It kind of hangs down because it's following the greater curvature of the stomach. About 45 centimeters long. You go from right colic flexure to transverse colon, and then when you get to the left side, it takes another turn called the left colic flexure or splenic flexure. When you went from ascending colon, that was retro, transverse colon is intraperitoneal. And then when you get to the flexure, again, splenic flexure, then you go back retro. Yeah.
Does that mean retro has sphincters?
Retro means it's behind the peritoneum. What do you mean sphincter?
Because the stomach to the duodenum or whatever, right, there's a sphincter.
Yeah, there is a sphincter in that example. But you don't always need a sphincter just because you're going retro. That's not always the case.
But there's some type of valve,
In the stomach there's a pyloric sphincter.
but not for retro or intro. Like the ascending colon, it doesn't have any type of valve.
No, not to my knowledge. Okay, there's no valve. But I feel like you really want there to be one. I'm not sure why. There's no valve.
Oh, because I mean, I thought retro means like backflow, because like, you know how there's the 3 phases in,
Oh, when I put retro, I mean retroperitoneal, not retropulsion. I think you're confusing my retro, so let me write retro. Because I did use a term called retropulsion when I talked about the peristaltic waves, but that was only for the stomach. And then there you do need a valve, so you don't, yeah. Okay. Yeah, sometimes students ask questions, oh yeah, that's why you're asking. Okay, I can see why you think this is a valve. Right, well anyways, after you get to the splenic flexure, then you just go down. They call it the descending colon. Let me, here's my notes on descending colon. From left colic flexure on down, descending colon. Now the descending colon is retroperitoneal. I have a cadaver presentation on Friday, and I'll show you the whole colon. And we haven't dissected out. We haven't left things retroperitoneal. Kind of cut it away from the back body wall so we can kind of pull it out a little bit so you can see the whole thing. Okay, so the descending colon is retroperitoneal. Now you kind of see when it starts to take a little bit of an S-shaped turn. That's when you start to call it the sigmoid colon. So it's curvy, and that leads directly to the rectum. So at somewhere between the sigmoid colon and the rectum, the tinea. Do you see how like there's hostra, and then the hostra disappear? That's because the 3 tinea coli, they restore the full layer, the full longitudinal layer of muscularis externa. Okay, somewhere at this juncture. So I'll put as a note on the board. Restore full longitudinal layer. Of ME, muscularis externa. So it happens at somewhere at the sigmoid rectal junction. Okay, and when you get to the rectum, that's it, that's the end. I mean, sigmoid colon is the end of the large intestine. So all those things are kind of like your journey through the large intestine, and all the way you're compacting the feces. Going up, across, and down, basically. A couple of pictures here. There's a picture of our torso back there. And do you see the transverse colon? I expect you to see that one. It's pretty obvious, I think. It's right there. So if I point to the individual pouches, it's like the hostra, but that ribbon, that's the tinea coli. So what's 526? Tinea coli. What do you think 526 527 is going for? Hostra. Those little pockets. Do you remember 491? Duodenum. What about 496? Main pancreatic duct. Okay. Therefore, what's 495? Pancreas. All right. Well, anyways, that's just some review. We have a lot of models and pictures. This picture is simply of the rectum, and it's the last 12, 15 centimeters. They call this the fixed terminal part. It's following the curvature of the sacrum. Although you can't see it from here. Okay, rectum. So fecal matter accumulates here, collects here. But you already knew that, right? I described the defecation reflex earlier. When you distend the rectal walls, the reflex is to contract and you push it towards the anal canal. The anal canal has an internal and external sphincter, and it's the external sphincter you can control with conscious thought there to help you hold it in, so to speak, until you get to the bathroom. Surrounded by external anal sphincter. The internal one is smooth muscle, the external one is voluntary muscle, skeletal muscle. So I listed the external one because it's the one you can control with conscious thought. I want to spend more time talking about the blood supply to the colon and the small intestine. So let's go back and start with the blood supply for the jejunum ileum. That would be superior mesenteric artery, shown here. Well, it's showing you a lot of things here, but focus on the superior mesenteric artery. Superior mesenteric artery, I'll just kind of list what it's supplying blood to here. As you can see, it's giving off branches to supply blood to jejunum ileum. And also colon, all the way up to the left colic flexure. Colon up to left colic flexure. Let me get a room on the board here. So that's it. That's a lot of intestine. That's most of the intestines, all the way up to the left colic flexure. So this the branches have names. You don't have to know them. Just be able to know that they're branches of the superior mesenteric artery, giving off all these branches that supply blood to the small intestines and colon there. And this model on the right shows it. There's the superior mesenteric artery right there. Okay, and all these cut branches are going to ileum jejunum. Then it shows you a good job of how all these branches here go to the colon, all the way up to this flexure here. That's the splenic flexure, the left colic flexure. The diagram, it's easier to understand. And all these branches supply blood up to the left colic flexure. So the inferior mesenteric artery will get the rest. This picture shows you the superior mesenteric artery, and all those cut branches of it, of the artery and the vein are there. This is the back side, so when you flip it over, this is what you see, that's the front. So I want you to look for those, the branches of SMA in the back. So kind of like when you flip that around and put it inside here, all of these cut branches are fitting those. Okay, that's what they're trying to show you there. So the inferior mesenteric artery will supply blood to the rest of the distal colon. They show you both. They're both branches of the abdominal aorta. Notice how the superior mesenteric artery, it kind of goes off to the right a little bit. And then the inferior one, it goes off to the left because it's got to go to the left side to supply blood to the distal colon, which is on your left. So I'll just say that the inferior mesenteric artery, it supplies, well, everything where you left off from, supplies from left colic flexure on down. That's everything after the left colic flexure, which is, like for example, if I say it supplies blood to the descending colon, would that be correct? Yeah, will be correct. And what if you say no, and in your notes you say it supplies everything from left colic flexure on down. Well, what does that mean? It means everything after that. So again, I teach concepts. I don't want you to memorize your notes. I want you to know what your notes mean. Here's a good picture of it. I already talked about superior. And there's the inferior supplying blood from descending colon, sigmoid colon, and all that. So I just put in a lot of pictures of the model next to the picture in the book. Okay, so my thinking in doing this is you study your book at home and you come to lab and you study these models which are in the classroom. And it's easy to not confuse superior and inferior because if you see both of them, you know the one on top is the superior one. And you know the one on the bottom that's closer to the bifurcation here, the aorta, that's the inferior one. It also, the other clue I gave you, it goes off to the left. You always see, I've seen that on every cadaver. It always goes off to the left. So again, what are the clues if it's not labeled? Well, that one's on top, so it must be superior. Also, identify this, green tube, duodenum. I did also say the superior mesenteric goes over the duodenum. It's another clue. Well, anyways, the other clue is you memorized what it supplies blood to. If it's going to here, that's inferior. It supplies the distal colon. Okay? And on this picture here, again, if you can see where the branches go to on this side, that's the inferior, that's superior. And that's the arterial blood supply. The venous drainage are part of what's called portal circulation. And the term first pass is often used to describe the portal vein because blood from the intestines must, quote unquote, first pass through what organ? Liver. Liver. Now the portal vein is shown at the top there. And I want you to know some of the veins that drain into it. There's portal vein right there. So notice how everything is going from a straight shot from intestines and spleen to the liver first, before you go into general circulation. There's no inferior vena cava here. Okay, this is all going to the liver. So what you have here is you have the superior. Let's go to this picture here. I kind of like teaching from this picture. It shows you the essentials of what I want you to know. You have the superior mesenteric vein. You have the inferior mesenteric vein here. Superior. Mesenteric vein. And this one is inferior. Mesenteric vein. Splenic vein is going to come from this side and going to merge with the inferior mesenteric vein. So this one is splenic vein. We're going to merge, and then we're going to merge with the superior one, and then go to the liver. So that's portal vein going to liver. Once again, blood returning from the intestines being drained by superior, inferior mesenteric vein, and also blood coming from the spleen. They're all going to merge, and all that portal blood will be processed by the liver. So this is kind of what we call portal circulation. It just goes straight from these structures to the liver before it goes back to the vena cava. So know what portal circulation is. Blood must, quote unquote, first pass through the liver. Before it is returned to systemic circulation. Systemic circulation. Okay. So on this picture here, they cut away the left lobe, part of the right lobe, so you can kind of see the portal blood entering the liver. But even though there's no directional arrows on here, blood is flowing to the liver. Portal vein along with proper hepatic artery. Okay, so blood is flowing into the liver. That's the dual blood supply. And let's remember the single venous drainage right there. All of these portal veins, hepatic veins. And there's inferior vena cava right there. Okay, so you first pass through the liver, you go in here, then out there. That's your liver, big filter. And I want to shift gears here and talk about the histology of the digestive tract, because there's a quiz on proctorial. I don't know if you've seen it. I want to get you ready for that. Now, the slides here, I don't teach from them anymore because I've taken other pictures from our collection since I first made these slides. But I've left them here. It's like, why delete them? They're good slides you can continue to study from. But I'm going to stop here and open those other slides. I posted these slides that I'm going to present now last night. I'll make a modification on my camera.